digestion and absorption. Within the human body, there's multiple enzymes that help us digest biological molecules present in our food. These macromolecules include carbohydrates, lipids, and proteins. Carbohydrates are broken down by carbohydrases, lipids are broken down by lipases, and proteins are broken down by proteases. Let's go into carbohydrates first. The main example of the carbohydrates is amylase, which breaks down starch into maltose. Amylase is produced from the salivary glands and the pancreas and is excreted to the small intestine and the mouth. Salivary amylase first hydrolyzes, for example, starch to maltose. Then, pancreatic amylase further hydrolyzes the remaining starch to maltose, and membrane-bound disaccharidases, maltases, then hydrolyze the maltoses into alpha-glucose molecules. Other examples of membrane-bound disaccharidases include sucrase, which breaks sucrose down to glucose plus fructose, and lactase, which break lactose into glucose plus galactose. Membrane-bound disaccharidase means that these enzymes are attached to the membrane of the epithelial cell of the ileum, which is the final part of the small intestine. Lipids are broken down by lipase enzymes, which catalyze the breakdown of triglycerides into monoglycerides and fatty acids. A monoglyceride molecule is a molecule with glycerol and one fatty acid attached to it. Emulsification is a process in which lipids are split up into tiny droplets called micelles by bile salts, which is secreted by the liver. The digestion of proteins are assisted with peptidases, also known as proteases. Endopeptidases hydrolyze the peptide bonds between amino acids in the central region of the protein molecule, and exopeptidases hydrolyze the peptide bonds on the terminal amino acids of the peptide molecules. These are the final parts of the polypeptide. Dipeptidases hydrolyze the bond between the two amino acids of a dipeptide. Dipeptidases are membrane-bound, which means they are found to the cell. They are attached to the cell membrane of the epithelial cell in the ileum. The absorption of triglycerides, after they have been emulsified by bile salts into micelles, is done by the process below. Micelles help move the monoglycerides and fatty acids after they have been broken down by lipases towards the epithelium. These micelles break down, releasing the monoglycerides and fatty acids. The monoglycerides and fatty acids diffuse across the cell surface membrane into the epithelial cell. As these molecules are non-polar, they can diffuse directly through the phospholipid bilayer without the use of a channel or transport protein. They combine to form triglycerides in the epithelial cell and associate with cholesterol and lipoproteins to form chylomicrons. Chylomicrons exocytose and enter lymphatic capillaries called lacteals. Chylomicrons pass into the blood after that. The triglycerides in the chylomicrons are hydrolyzed by an enzyme in the endothelial cells of blood capillaries, from where they diffuse into cells that require monoglycerides or fatty acids.